Thanks very much. I'm delighted to be here. We've had a pretty high-profile year, and I'm very pleased to present the company because this has really been a miraculous time for us, converting from a really small company to a company with some very interesting prospects. So we call ourselves unashamedly the leader in T-cell receptor T-cell therapy, and I'll try to give you some reasons why that is a perfectly justifiable claim as I go through the presentation. What do? We engineer, and that's the most important word probably in the presentation, T-cell receptors for autologous T-cell therapy in cancer. We've got great clinical data. We have complete responses not only in myeloma, but also in a solid tumor, sarcoma. We've got a very long pipeline of validated targets, which you can't approach with other types of treatment. We've just signed a big deal with GSK in June 2014. We've also, even more recently, just signed a Series A funding entirely here in the USA, where we raised $104 million. And that is basically because we have a strategy to build a major oncology franchise of proprietary products, and that's what the funding was about. So just a little word about the origins of the company. I founded the company in 2008 as a spin-out. We founded it as a virtual company with Carl June as our collaborator in the University of Pennsylvania in Philadelphia. He was unbelievably helpful. We provided the products, and he held the IND and provided the cell manufacture, the vector, and the patient. So it was a fantastic collaboration. Over the last 3 years, we've moved away from that virtual model and become a fully independent operational company, partly because we needed to do phase 2 trials outside the Penn context. And in the last 3 months, as I say, we've signed a big deal with GSK and the Series A funding round. So we have a base in Philadelphia, a clinical and regulatory base headed up by Gwen Binder-Scholl, who spoke on the panel this morning. And I think it's already out of date. I think we're 15 people there, and we're 40-something in Oxford in the UK. The original technology was spun out of Oxford University, which is why we ended up there. So we've ended up with a very impressive board, I think, as a result of the Series A round. We have our existing chairman, Jonathan Knowles, who's probably very well known to some of you, ex-research director both at Roche and at GSK. We've been joined by David Mott as our Series A investor, lead investor, who was CEO of Adaptimmune, and we have Elliot Siegel, who is former vice president of R&D for BMS. So I think we've got a pretty solid board in place. So what is so special about this technology, and why do we have so many targets? Well, really it's because there are 2 ways you can identify a cancer cell. You'll all be familiar with the antibodies targeting the cell surface proteins, but actually, and that's of course what CAR T-cells in the T-cell therapy space also target. But actually, although they're wonderful targets, there aren't very many of them, and in industry terms, the industry is actually running out of targets. So we are targeting something completely different. We're targeting the same proteins, but in their broken down form. A tiny fragment, usually 9 amino acids long, is presented on the cell surface on the HLA molecule. So we call them HLA peptide antigens, and that is what a T-cell receptor normally targets in man, and that's all we're interested in. No one has managed to manufacture antibodies to recognize effectively these, and that's why we have so many targets, because unlike the antibody world, there are hundreds of these targets known in the literature, and in fact we have around 28 validated targets in-house as I speak. So what's the process? On the left-hand side, you have a patient. The patient is screened for both the HLA and the antigen. We take an apheresis with the T-cells. We transduce the T-cells back with the T-cell receptor with the right specificity using a lentiviral mechanism. And then we expand the antigen-specific T-cells using CD3, CD28 beads. So I can now give a plug to 2 of the other speakers, because those beads, of course, are LifeTech Thermo Fisher beads, where we have an exclusive license for all T-cell receptors for those beads. And the other plug I can give is that PCT, or Neostem, has just presented, is our manufacturing base to produce this cells back for the patient. The patient then undergoes conditioning lymphodepleting chemotherapy, but it's not therapeutic chemotherapy, and then the dose is administered in an IV infusion. So we start with a target and how to identify it. We spent a lot of work doing that over the years, and we end up with a system where we analyze the targets into the extent to which they appear on cancers, which is in the sort of pale blue box at the top of this. So this one, something called STEAP1, is on all of prostate, most of renal, etc., etc. But then more importantly, we then do a screen to see whether the target is also on healthy tissue. And at the bottom of this, for those of you who can't see, are actually 72 bars. Each one represents a healthy tissue, so something you wouldn't want to target. The most dangerous are on the left, so sort of cardiovascular, lung-type tissues, and then immune-privileged sites are on the right, such as testis, which doesn't produce HLA. And we produce this chart to show the extent to which a T cell would be activated and kill the cells. Now, this was obviously, I show this because this would be extremely dangerous because it has a large presentation in the red bars of most dangerous tissues, so we would never take this into the clinic. But the reason I show this is because, in contrast, this is our clinical candidate using exactly the same scale, using exactly the same basis, NY-ESO, and you can see this is a member of the cancer testis family, that you can actually generate extremely clean targets. They tend to have slightly lower presentation in individual cancers, so you can see here it tends to be between 30 and 70% of cancers. But the beauty of this is that you can target it and you shouldn't have an effect on normal tissue, and that's what we found in the clinic. So I said right at the beginning that engineering T-cell receptors was very important to us, and the reason is really it comes from the problem of thymic deletion in nature, which effectively deletes all T-cells of high affinity that look too similar to self, and of course most cancer antigens look too similar to self. And in fact, we published this some years ago in 2012. If you actually look at the affinity of a T-cell receptor in nature, so these are just taken from random donors, for cancer versus virus T-cell receptors, you could see that the affinity on this chart is much higher for virus TCRs, which have not been deleted because they don't look like self, than for cancer. All the blue ones are cancer T-cell receptors, and all of the red ones on this log scale are for viral T-cell receptors. So all we do, which sounds easy but took a lot of many years of engineering expertise, is we convert the low affinity T-cell receptors up, not hugely, but to the range where you see the viral TCRs for that specific antigen. So why does that matter in practice? Well, it matters because you need quite a lot of copies of the antigen on the cancer cell to activate a T cell, generally between 100 and 500 copies before you actually get full activation of a normal T cell receptor. And so and guess what? Cancers actually present between about 10 copies and 100 copies on the cell. So what we're trying to do is to overcome that problem, and the way we do it is through a proprietary phage display system, which produces what we call a barcode panel of many, many different affinities. And what you find is that you convert something, this is an ELISpot assay, which is showing absolutely no reactivity to a cancer cell because of this downregulation problem I mentioned earlier. What you find is as you go up the affinity scale, you hit a position where you can actually see each brown dot representing activation of a T cell. You can see excellent recognition. And in fact, these T cell receptors that we produce will recognize down to between 5 and 10 copies on the tumor cell. So that's the philosophy behind the company, to engineer effectively blind T cells into T cells that actually can see the cancer. We're in the clinic with an extensive program with our NY-ESO. The red arrows indicate the indications we're already in: ovarian, multiple myeloma, melanoma, and sarcoma, and in planning is non-small cell lung cancer, so it's a dotted red line. We've dosed 36 patients, and there's some published data, in fact, previously with our construct at the NCI in the JCO 2011 in sarcoma and melanoma, also using this construct. So it's been through a reasonable number of patients. It's at a very good set of centers throughout the United States, through the NCI, Yale, CHOP, etc. Many of the centers I'm sure that you'll recognize in the cell therapy world. And as you can see, there's one center in the UK, which we hope will start an esophageal study soon. So what happens? The general study design, as I said, is you take an apheresis. It takes roughly 28 days to manufacture the T cells. A week or a few days before the administration of the cells, you have the cytoreductive chemotherapy, and then you have a single infusion, which is somewhere between 1 and 10 billion cells, and then you take response assessments at 1, 2, and 3 months. We've dosed quite a number of patients now. 27 in myeloma have been treated, and 15 patients with solid tumors in total. And I'll just show you data from 1 or 2 patients, but we will be announcing the presenting, or Crystal McCall from the NCI will be presenting the sarcoma data later this month at a conference in Berlin, a sarcoma conference. So the reason that we believe in the efficacy of these T cells is this is a comparison between a meta-analysis of all stem cell transplant results from multiple myeloma on the left, around 24%. You can improve that with Velcade to 38%, and our running response rate is about 73%. And in sarcoma, I normally just present this one picture just to show you the extraordinary effect of T cells in getting a complete response with a patient, obviously riddled throughout her lymphatic system. And that complete response on a PET scan completely took away the cancer. That's a complete response. Just a couple of words about the company itself. So we've heard quite a few things in the plenary session right this morning, in fact, about there is an enormous number of questions still to be answered. The whole regulatory pathway, clinical pathway still has to be defined. We're looking very carefully at the exact clinical setting for these T cells. There's been quite a lot of discussion about how you optimize the cell product for durable and reproducible efficacy. We've been obviously very concerned to do that. There's a whole question of cost of goods, the optimization of production for cost-effective commercialization, and indeed there's the question of what type of diagnostic would you use in this field. And on top of that, we have an enormous long pipeline of products. As I said, we have 28 targets which are validated in the cupboard at the moment. Every single one of those is a multi-cancer product, so you can see it's rather a long pipeline. So we decided to form a strategic deal with GSK. It is a strategic deal. It's not just a license of an individual thing. We are looking at not only getting their help in pushing forward our existing clinical plan, NY-ESO, to the market as quickly as possible, but we have complete programs in looking at the whole clinical regulatory aspect, the clinical aspect, the CMC optimization, and GSK has a lot of in-house expertise actually in lentiviral production, and of course the companion diagnostic. And very importantly for us, we are carrying out all of that work. They're paying for it, and then we have all of the IP for that to apply to our own pipeline. So it is a real collaboration, looking at how to get the best type of cells, how to commercialize them, and then how to produce our own pipeline with all of that IP. And also it's quite handy financially. We get up to $350 million payable over the next 7 years from this June to achieve the above. And after that, in fact, that we'd have further milestones and, of course, royalties on any product which comes out of the collaboration. We are looking at some other T-cell receptors with them, and the net result is that they will have a pipeline of T-cell receptors, but we will still retain the vast majority of that 28 targets that I mentioned earlier. So we just completed our Series A funding. We actually set out to raise $25 million, but we ended up with $104 million. And that's really important to us because it's designed to fund our proprietary products through the clinical trials. And just as important as the amount of money, in fact, was the quality of the investors. I think you'll agree that that list is a who's who of US investor base, not only venture capital firms, but public company. And that's because we're very, very interested in the strength of the shareholder base to take us through whatever the vagaries of the stock market or anything else. We have an enormously strong shareholder base with which to keep the company well funded to achieve what are very ambitious targets. So in summary, I think the reason that we achieved such a deal is there are a lot of core differentiators between us and everybody else in the field, starting at the bottom left of that red box with the antigen identification. We can then make the T-cell receptors. We can engineer them. We've got a lot of IP ourselves on lentiviral vector production. We can actually do the T-cell expansion with those Thermo Fisher LifeTech beads that I mentioned earlier, and we've got a lot of clinical regulatory experience in this country with which we're obviously planning the whole pipeline. So in other words, it's a complete sort of one-stop shop for producing a pipeline of T-cell therapies in this country. So in summary, I think we're very well funded, a lot better funded than we were in June, I can tell you that, both through the GSK deal and by this list of what I regard as the sort of top quality US investor base. We're backed by the strategic deal with GSK, which I think is giving us an enormous help while we're doing all the work. We will have a very strong news flow. our own product, our own proprietary multi-cancer candidate, not NY-ESO, will go into the clinic in 2015, and we're already started work on the further pipeline, and we will keep rights over those programs. So thank you very much indeed.